Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss NADH shuttle system. What is NADH shuttle system? So we know that respiration can be of two types. One is aerobic respiration, second one is anaerobic respiration. Aerobic respiration produces more energy than anaerobic respiration. During the aerobic respiration, eukaryotes produces NADH and FADH2, which is generally produced through glycolysis and Krebs cycle. Glycolysis occur in the cytosol and Krebs cycle inside the mitochondrial matrix. So the NADH and FADH2, which are produced during this process, ultimately convert into adenosine triphosphate following electron transport chain and ATP synthesis. So the mitochondrial NADH and FADH2 can easily move to the electron transport chain, which is present on the inner mitochondrial membrane, and finally convert into the adenosine triphosphate. But the cytosolic NADH, which is produced during glycolysis process, have to move into the mitochondria and to inner mitochondrial membrane to perform electron transport chain and to produce adenosine triphosphate. So for this, the cytosolic NADH have to transfer into the mitochondria. But the cytosolic NADH is impermeable to the inner mitochondrial membrane. So it will follow an indirect pathway or a specific pathway called as shuttle system. So this shuttle system facilitates the cytosolic NADH to move into mitochondrial matrix. There are two shuttle systems. One is glycerol phosphate shuttle system and malate asparatate shuttle system, which are present inside our body. There are two shuttle systems have been recognized. One is glycerol phosphate shuttle system. Second one is malate asparatate shuttle system. So what is glycerol phosphate shuttle system? Glycerol phosphate shuttle system, actually this shuttle system is controlled by two molecules. One is glycerol 3-phosphate, second one is dihydroxyacetone phosphate, and two enzymes control this reaction. One is NAD linked cytosolic glycerol 3-phosphate dehydrogenase, and the second one is FAD linked mitochondrial glycerol 3-phosphate dehydrogenase. So at first, in cytosol, reducing equivalent from cytosolic NADH plus H plus is transferred to glycerol 3-phosphate via dihydroxyacetone phosphate. So from NADH H plus, reducing power or reducing equivalent move to the glycerol 3-phosphate via dihydroxyacetone phosphate, and the enzyme which helps in this reaction is NAD linked cytosolic glycerol 3-phosphate dehydrogenase. Finally, glycerol 3-phosphate enter inside the matrix, where it converts into the dihydroxyacetone phosphate again, reducing the FAD into FADH2. In this way, the reducing power enter into the matrix. The reducing power finally convert the FAD into FADH2, and that FADH2 again enter into the electron transport chain to produce adenosine triphosphate. So it is a two-step reaction. At first, the dihydroxyacetone phosphate collecting the reducing power from NADH plus H plus converts into the glycerol 3-phosphate. It transfer via inner mitochondrial membrane and move into the matrix, where it again convert into dihydroxyacetone phosphate, reducing FAD to FADH2. FADH2 enter inside the electron transport chain. That dihydroxyacetone phosphate again move back to cytosol, and that FADH2 enter inside the electron transport chain to perform the electron transport and to produce adenosine triphosphate. Here two enzymes control the reaction. One is NAD linked cytosolic glycerol 3 phosphate dehydrogenase and the second one is FAD linked mitochondrial glycerol 3 phosphate dehydrogenase. C means cytosolic and M means mitochondrial. The next one is malate aspartate Shuttle system. So here two enzymes control the whole reaction. One is malate dehydrogenase and the second one is aspartate aminotransferase. These two enzymes control the whole reaction. The first step is in cytosol after glycolysis, malate collects the reducing equivalent from NADH plus H plus via oxaloacetate, which is catalyzed by cytosolic dehydrogenase, malate dehydrogenase or MDH. Oxaloacetate collects the reducing power from NADH and H plus to form the malate. Malate enter inside the mitochondria where it convert into oxaloacetate, and in this process NAD plus is again converted to NADH plus H plus. During the movement of malate, one antiporter helps in this movement, that is malate alpha ketoglutarate antiporter TA. Then inside the matrix, malate convert into oxaloacetate, finally forming the NADH plus H plus, which enter into electron transport chain. Then in the mitochondrial matrix, oxaloacetate is formed, which is impermeable to inner membrane of mitochondria, so cannot be transported back to the cytosol. Hence it combines with glutamate and undergoes transamination reaction to form alpha ketoglutarate and aspartate. Alpha ketoglutarate and aspartate is formed when oxaloacetate combines with glutamate. Oxaloacetate inside the matrix combines with glutamate to form aspartate and alpha ketoglutarate. Okay, so it is the step 3 reaction. So at first oxaloacetate convert into malate in cytosol, it enter into matrix where malate convert into oxaloacetate. Oxaloacetate combines with glutamate, form aspartate and alpha ketoglutarate. Then alpha ketoglutarate and aspartate again exported to cytosol using the antiporter malate alpha ketoglutarate antiporter. That alpha ketoglutarate and aspartate again transported into the cytosol. Okay. Then in cytosol, alpha ketoglutarate and aspartate again transaminated to form glutamate and oxaloacetate, and that oxaloacetate again perform another malate aspartate Shuttle system. Alpha ketoglutarate and oxaloacetate, alpha ketoglutarate and aspartate again in cytosol form glutamate and oxaloacetate by using aspartate aminotransferase enzyme. So here two enzymes works. One is your malate dehydrogenase MDH masala, and the second one is aspartate aminotransferase, and two antiporter works like malate alpha ketoglutarate antiporter and glutamate aspartate antiporter. Malate alpha ketoglutarate antiporter is TA and TB is your glutamate aspartate antiporter. So it is all about our two Shuttle system which are present. So with this I am concluding my lecture video here. If you like it, please hit the like button, share with your other friends and colleagues, and please subscribe to my channel Sunanda's Tutorial to get more important videos like this. Thank you.